I hand over you the session, sir.
Thank you, madam. Thank you. Thank you very much, madam. Uh, my voice is clear, madam?
Yes, sir. Very good, sir.
Yeah. Slides. Good afternoon to everyone, madam, who are online and offline in this wonderful training program.
Good afternoon,
Organized,
sir.
good afternoon, madam. Organized jointly by JNTU Kakinada and also Vijaya Institute of Pharmaceutical Sciences for Women, Vijayawada. And thank you, Vani, madam. You have introduced nicely. I'll be speaking on two topics which Bhavani madam and you have communicated that I have to speak for 2 hours, 2 to 3 and also 2 to 4. The first topic I'll be covering on the pharmacological evaluation and safety studies of the herbal drugs and regulatory requirement. We are working more than 2 decades in this line of area, and what are the herbal drugs and what are the botanicals and what are the regulatory affairs to carry out the herbal products as drug and also the nutraceutical point of view. I'll be covering several aspects and the terminologies that is well confused and also to be placed in the to get the approval of the drug. What are the terminologies that are used and how this data entry and also documentation of this data at the laboratory level and also to the regulatory agency and how to get it approved in this theme of presentation. I'll be covering the topics, and before going in depth of the research in the topic, I myself is Dr. C.H. Vilas, Chief Scientist and Head Pharmacology. And I worked in Sri Vishnu College of Pharmacy before I joined 2 decades before in NBRI. And working in this line, and we have worked out several products. And in this line, this is a premier institute of CSIR, and more than 100 scientists are working in several disciplines in this organization, and basic science to the application oriented. Most of the scientists are background is botanical and also the pharmaceutical chemistry, and the chemistry background biotechnology, molecular biology, and the tissue culture and herbarium, and we are it is covered in more than 200 acres of the land of our institute. And the topic which I have covered, which I have projected in this presentation, will be, as Madam indicated, the pharmacological evaluation of herbal drugs and regulatory affairs. And the terminologies which are more to be focused and kept in mind is what are herbal medicines. Herbal medicinal products include herbs and herbal materials, herbal preparations, finished herbal products containing parts of the plant and other plant materials, or combining thereof as an active ingredients. These helps include a crude plant material that may be obtained from the leaves, flowers, fruits, seeds, and even on the stems. And which part will be used for the product in the making of the medicine as an herbal medicine. It not only covers the plant material, it also covers the addition of herbs or the fruit juices or a gums or a fixed oils or essential oils, resins and dry powders of herbs. These herbal preparations are the basis of the finished herbal products and may include comminuted or powder herbal materials or extracts or tinctures or fatty oils of herbal materials. The finished product consists of the herbal preparations made from one or two more herbs. Although the herbal medicines are very popular in the society, only few herbal medicinal herbs have been scientifically validated. And based on this concept, more data and more robust data is to be generated to evaluate the herbal products from the raw material to making to the medicine form. And the safety is not proved and the quality is not assured. When we look into the European and other agencies which is to be exported, to address these issues, US FDA has created a separate guidelines document of the for botanical products for NDA and IND submissions. And similarly in India, CDSCO, because most of the presentations I have seen that they have covered CDSCO and DCGI, but I'll give you a glimpse of CDSCO also in this presentation. And there is a separate guidelines for the botanical products called as phytopharmaceutical guidelines. And in 2013, and the first revision came in 2015. And when we look into the Minister of Health and Family Welfare, the Drugs and Cosmetics Act 1945, and several amendments came from subsequently to the 2015. And it is clear in the website. If anyone are interested, they can see on the Ministry of Health and Family Welfare site, which it is gazetted, and the documents can be downloaded and followed the guidelines. And when we look into the Central Drug Standard Control Organizations, CDSO, the Drugs and Cosmetics Act 1940, and it's not only focused on the herbal products and also the synthetic and the bulk drug industry, which the molecules for the synthetic origin or the drugs, how it can be regulated, what are the norms to be followed, how the consumer accepts the product of the finished product by looking to the quality and also the efficacy and safety point of view, and what is the prescribed drugs and the dosage forms to be clearly mentioned on the label, and how this documentation came from the laboratory level to the finished product to be placed on the bottle or a product. These are the things which has to be followed strictly, following the Central Drug Standard Control Organization. This is the site which can be referred for the future and advanced in depth of the documents which can be downloaded from the site. When we look into the Drugs and Cosmetics Act, because we are working on the herbal products, because our premier plant organization works on the plant products and plants. When we look into the plants, that is when we look into the herbal products, herbs, plants, medicinal plants, ornamental plants, decorations, and the pollution control plan oriented, how we in-house, and what are the plants that are made into the botanical garden, and how this is conserved. These are the classical. When we look into the medicinal point of view, we'll be focusing much on the medicinal plants. The ancient texts like Ayurvedic texts, Ayurveda, Siddha, and Unani medicines, under section 3A, classical formulation described in specific texts in Ayurveda. When we look into the patent or the proprietary medicine, new formulation based on the medicinal plants, it is clearly mentioned when it is looking into the schedule 1. And the Aushguna, most of the plants in Ayurveda, when we look into the most of the aqueous or hydroalcoholic extracts are used. When we look into the aqueous and hydroalcoholic extracts, what are the norms to be followed, and what are the specific guidelines that has been mentioned, that has been documented. Based on this document, the presentation will be much on the phytopharmaceutical mode of the drug development. As per the Gazette in the rule of the Drugs and Cosmetics Act 1945, that is includes the purification and standardization fraction with the defined minimum 4 active phytochemical compounds. That is, for example, if a plant has been extracted or fractionated and subsequent fractionation, that may be a biological standardization or a chemical standardization and the microbial standardization. These, when we look into the chemical standardization, the bioactive compounds are the specific chemical markers to be quantified, both on the qualitatively and quantitatively, of the individual extracts of the plant material that may be used either for the internal use or external use. When we look into the internal use, most of the drugs of the herbal were given orally, and when we look into the herbal products that has been processed either in the for the human beings or the animals, for diagnosis, treatment, mitigation, or prevention of any disease or disorders, does not include administration by parenteral route. But we have to keep in mind that these plant products are given orally but should not be administered by parenteral route. Based on this background, we will go into the attributes like manufacturing license or marketing authorization, the product license as Ayurvedic medicines made using botanicals as activities, as proprietary Ayurvedic medicine issued by licensing authority of the state in which the manufacture is undertaken. The product licensed as a new drugs at the phytopharmaceutical mode as a new drug whose import of the market is marketing is authorized by the DCGI, as I shown that CDSCO. How many herbs, when we look into the products in Ayurvedic medicine, can be single or a combination of more than one, with each ingredient being listed in the other in the text, which in like Ayurveda or Unani or a Siddha, these plants should be mentioned clearly on the texts in the Ayurveda. First schedule or processed materials including extracts as permitted under the Section 4 of the Drug Controller Authority of Regulatory (DCR). When we look into the phytopharmaceutical mode, recommended as the phytopharmaceutical for a single botanical to begin with, combining more than one is not bad, but not need a lot more scientific data on the rational interactions and other aspects which are much more difficult to generate. The source of the botanical has to be mentioned in one of the authoritative texts of the Ayurveda as per schedule of the DCR, that is Drug Controller Authority of Regulatory, because it is clearly mentioned in which text it has been mentioned as the plant material has been for what purpose and what intended the plant, and what is the plant material is processed and making it into a medicinal form. This issue should be clearly mentioned in the text of Ayurveda. When we look into the products in the modern era in the phytopharmaceutical mode, can be a botanical from any other. It no need to be mentioned in Ayurveda. It is mentioned or not mentioned. Can be any botanical form, any part on the globe. It is combined with a mineral or a metal permitted as per the desire. The plant material can be mixed with a mineral, can be mixed with the mineral or metallic forms in the Ayurvedic text as permitted. And when we look into the phytopharmaceutical mode, these are not permitted in the definition of the phytopharmaceutical, and no provision is placed here. When we look into the extracts of the botanicals, extracts of the herbs as per schedule, chapter 4, as per to applicable to Ayurveda. When we look into the phytopharmaceutical mode, because the presentation is more focusing on the plant and the phytopharmaceutical molecules and the drugs, that is a purified and fractionated extracts that meet the definition of the phytopharmaceuticals and need to be supported by more scientific, robust data on the evaluation of this medicine coming from the phytopharmaceutical regulations. And the data to be generated to conduct clinical trials or import and manufacturing of the phytopharmaceutical drugs in the country. What is the data to be generated to be generated before going to the clinical trials, and or import or a manufacturing of the drug as per the norms of the phytopharmaceuticals in our Indian scenario. That is identification. We have to identification of the plant material, authentication and source of the plant used for extraction and fractionations, and process for extraction and subsequent fractionation and purification, and formulation details of phytopharmaceutical drugs, how it has been formulated, and what are the formulation details to be given very clearly, and manufacturing process of formulation, and stability data and safety and pharmacological information. Because when we look into the process of the in the pharmaceutical point of view, we have been in depth of the study of more than several disciplines in this covered, from the pharmacognosy, from the identification to authentication, and the chemistry, followed by the marker identification and quantification of the markers, both in the qualitative and quantitative, or the specific 4 markers to be identified, and how it is processed and the formulation made, whether it is a tablet or a capsule or ointment, pharmaceutical dosage forms to be made. And in this, the stability studies has to be made. What is the expired date and the manufacturing date to be placed on the sheet of the product, and the detailed safety, either it is a toxicity or a subacute toxicity or a chronic toxicity, and the safety studies to be mentioned with efficacy studies in depth in the information to be given to the regulatory authority to get it approved. When we look into the IND filing of phytopharmaceutical drugs, data to be submitted along with the application to DCGI, data generated by the applicant, that is identification, authentication, and source of the plant used for extraction and fractionation. In this, under this heading, we have to cover the taxonomical identity, morphological and anatomical description, because pharmacognosy plays a key role in the plant products development in the pharmaceutical line of the discipline of the education. Therefore, the taxonomical identification, morphological, anatomical description, confirmation, and confirmation of identity and authenticity of the plant material to be available. The natural habitat and geographical distribution of the plant, because the plant that may be available in Andhra Pradesh that may not be available in Uttar Pradesh or in Delhi. What is the natural habitat, in which area and where it is located? What are the geographical, where it is distributed, and where it is collected, whether it is a collected variety or a wild variety is used, and how do you standardize that plant material, and what is the season and the time of collection, what is the source of the plant, including its geographical location and season or a time of collection, statement indicating whether the species followed the, and whether it should not come under the endangered species as a restricted under the Biological Diversity Act 2002, or any other genotype or a chemotype or ecotypic variability of the species to be noted, and followed strictly the Biological Diversity Act 2002. The list of the grower or a supplier, because if it is we are using on the material that has been supplied from the trading point of view, the information of the harvest location, growth condition, stage of the plant growth at harvest, harvesting time, collection, washing, drying, storage, handling, and garbling and transportation, grinding, pulverization of the plant material, sieving for getting uniform particle size of the powdered plant material. These are the data to be generated by the applicant before submitting to the regulatory authority. And when we look into the IND filing of the data to be submitted, not only on the part one, there is a quality specifications, namely foreign material, total ash, acid insoluble ash, pesticide residues, what are the pesticides that are present, and whether it is used, and what are the limitations of the heavy metals, what is the microbial load of the product, what is the, how do you identify your product is a, therefore the chromatographic fingerprinting profile should be marked with the phytochemical reference marker on the what is the assay that we followed for the bioactive or the phytochemical compound, and the chromatographic fingerprinting of the sample as per the test method given for the quality control of the phytopharmaceutical drug. And undertaking to supply of the plant duly labeled and a photocopy of the certificate of identity confirmation issued by the qualified taxonomist, along with the drawing or photographs of the diagnostic morphology and histological features of the botanical raw material used for the confirmation of authenticity. Then the processing of extraction, purification, the quality specification and testing methods of starting material, and steps involved in the process of the solvent, what is the solvent used, what is the extract value, what is the solvent residue test, and the phytochemical test and microbial load in the heavy metal contamination, and the chromatographic fingerprinting profile, and the phytochemical reference markers, and characteristic markers of the constituents, characterization of the final purified fraction, data on the bioactive constant of the final purified fraction. And when we look into the data of packing of the purified and characterized final product, storage conditions and labeling, when we look into the formulation of the phytopharmaceutical drug applied for the details of the composition, for example, if the plant is not only the single plant, it has been mixed with the different ingredients and different bioactive fractions, that comes under the category of the composition. What is the proposition of the final purified fraction with the defined markers of the phytopharmaceutical drugs as per unit dosage, name and proportions of all excipients, what are the excipients that have been, what is the percentage of the excipients, what are the stabilizers if they have been used for the increase the lifespan of the product or expiry date of the product, what is the stability condition of the product, of the packaging material, and the test for identification of the phytopharmaceutical drug, and the quality specification for active or inactive phytopharmaceutical fingerprinting, fingerprint profile with the phytochemical reference marker, assay of active constants or characteristic chemical markers, and manufacturing process of the formulations under this six is of heating, the outline of the method of manufacturing of the dosage form, that may be a tablet or a oral dosage form or other dosage form. In the oral dosage form, most of the herbal drugs we use the oral dosage forms, that may be in the tablet or a capsule or a compressed form, along with the what are the environmental controls that have been used in process of the control tests and limits for acceptance, details of all packing materials, packaging steps, and description of the final packs, and the finished product quality specification including test specific for the dosage form, quality, and the chromatographic fingerprinting profile with the phytochemical reference marker, and assay of active constants and characteristic marker if active constants are not known. When we look into the stability studies, the stability data of the phytopharmaceutical drug described as 4 above stored at room temperature, at the humidity under standard conditions, at the zero at the starting initial level, and also the 1 month, 2 month, 3 months, and 6 months. The stability data of the phytopharmaceutical drugs in dosage form of the stored at the specific temperature and the standard humidity conditions for up to the level of 6 months in a packaging intended for marketing. When we look into the safety and the pharmacological information, data on safety and pharmacological studies to be provided, and this is the main area which we are in the pharmacological point of view, the safety that may be acute, subacute, or chronic toxicity, the animal toxicity and safety data 28 to 90 days repeated dose oral toxicity, that is clearly mentioned. It should be carried on 2 species, that may be in rodent and also in the non-rodents. and in vitro genotoxicity data, Ames test and chromosomal aberration test should be performed as per the Schedule Y, and dermal toxicity test for the topical applications, and teratogenicity studies also to be carried out, intended to be used for the during pregnancy. Based on this data, it goes for the human clinical trials. The clinical trials of the phytopharmaceutical drugs to be conducted as per applicable rules and guidelines for new drugs. For all phytopharmaceutical drugs, data for phase 1 to be determined maximum tolerated dose and also associated toxicities. And the protocol should be submitted prior to performing the studies. The data of results of those finding studies performed and the protocols should be submitted prior to performing the studies, provided that in case of phytopharmaceutical drug already marketed for more than 5 years, where there is adequate published evidence regarding the safety of the phytopharmaceutical drug, the studies may be abbreviated, modified, or relaxed. Therefore, from this we can understand that the phytopharmaceutical drug, if the herbal drugs are to be marketed or imported or exported to be comes under the category of the phytopharmaceutical drugs, there is an relaxation if the drug is already been used for a long period of more than 5 years. The in confirmation of the clinical trials, submitting protocols for approval for a specific or a specific safety efficacy study proposed specific to the comes under the phytopharmaceutical drug, submitted proposed protocol for approval for human, because every institute or the R&D organizations, both the ethical clearance to be obtained, both at the human applications and also the animal applications, and at the IHAC, this institutional level protocols to be approved. And what is the how do you validate, and these validated protocols to be covered under the safety and efficacy data to generate the pharmaceutical dosage form, the product as per the rules and guidelines. The submitted information has to be maintained during the above studies. And the regulatory status, the status of the phytopharmaceutical drug marketed in any country under in any country under any category like functional food or a dietary supplement or as a traditional medicine as an approved drug. These are because there is a terminology like functional food, dietary supplement like nutraceutical supplements, or the traditional medicines are being used for a very long time. And when we look into the marketing information, the details of package insert or the patient information sheet of the phytopharmaceutical drug to be marketed, draft of the text for label and cartoon. These are to be where it is to be marketed. When we look into the products in the distribution of the product in the state level and manufacturing unit, we have to get approval from the state level also. And the post-marketing surveillance, that is PMS, the applicant should furnish periodic safety update reports every 6 months for the first 2 years after approval of the drug is granted. For subsequent 2 years of the periodic safety update reports need to be submitted annually, because post-marketing surveillance is more important. When we look into the teratogenicity studies, the drug is used mostly for the specific point of view, but it is having some other adverse effects or the beneficial effects, and this comes under the surveillance, which after marketing, that is comes under the post-marketing surveillance. Initially is to be recorded for the 6 months, the every 6 months for the first 2 years to be reported, and subsequently after the 2 years periodic updates to be made annually. Any other relevant information which the applicant considers that with the help of the scientific data or the observation has been made, that has to be covered under this application. These under the headings of 14 subheadings under the phytopharmaceutical drugs under the DCGI. These studies has to be carried out in at the laboratory level. And these herbal drugs, when we look into the phytopharmaceuticals more, there are the specific guidelines to be followed by FSSAI, that is Food Safety and Standard Authority of India, that is guidelines for the food supplements and nutraceuticals. But when we look into the guidelines of the food supplements and nutraceuticals, under the regulations of 2015, published in on November 2016, food or health supplements or nutraceuticals, food for special dietary use, food for special medical purpose, functional foods. These are the 5 areas which is covered under the regulations under the food safety and standards under the regulations of 2015. And in these regulations, if we see FSSI guidelines, there are the specific plant extracts, what is the percentage of the extract to be used, and what is the name of the plant, and what is the botanical ingredients, what are the vitamins and minerals or trace elements, and what are its limitations that has covered in the products to be comes under these regulations, and what are the amino acids and other nutrients, what are the food additives to be used, and even the probiotics and the prebiotics, the microorganisms which is like lactobacillus also has been covered under this FSSI guidelines. This FSSI guidelines, the Food Safety and Standard Authority of India. Under this FSSI guidelines schedules, when we look into the content, list of vitamins and minerals and their component, in this under this category there are 15 vitamins and 15 minerals at the permissible doses has been given, and a list of amino acids and other nutrients. There are 11 essential amino acids, 14 non-essential amino acids, 4 nucleotides has been covered under these guidelines, and their limitations and what is the permissible limit of their usage, and a list of vitamins, minerals, trace elements allowed to be used in food, that is vitamins and minerals. The number is mentioned. List of the plants or botanical ingredients. Under this guidelines, there are 400 botanical plants listed along with their permitted range of use. For example, if the plant is toxic or a poisonous, but the plant to be used as a medicinal purpose as per the classical text in the Ayurveda, and how it is shodhana or how it is remove the toxicity of the plant, and what is the percentage of that plant to be used. These are the along with the permitted range. It should be labeled on the content, and list of food additives for health supplements, nutraceuticals, for special dietary use, for special medical purposes, for special medical purpose for weight reduction. For most of the obesity drugs in today's scenario, this is hypolipidemic, and several weight reduction drugs of plant origin and natural origin has been much focused to be used on the good manufacturing practice levels, to be used in the formats such as the tablets or the capsules that is recommended for the oral use. Then the name of additives and permitted level mentioned to be given on the information. On the label, list of ingredients as in nutraceuticals. When we talk about the nutraceuticals, botanical ingredients are different, and the list of ingredients as nutraceuticals are different. List of pure natural products or extracts with marker content and their purity criteria permitted range. There are 187 ingredients has been listed. When we look into the list of strains as probiotics, there we see the number here, that is microorganisms listed. When we look into the prebiotic compounds, there are 13 compounds has been listed. What is the media that helps in the growth, multiply the bacteria, that is a probiotics. That is a combination of a prebiotics and a probiotic, that is symbiotics. In today's scenario, most of the strains that are used in the gastrointestinal infections or in the diarrheal conditions, most of it is recommended use of the strains of probiotics along with the prebiotics. And when we look into the development of our evaluation of IND and NDA filing, what are the steps that has been covered under the CMC, that is a chemical manufacturing and new drug application, chemical manufacturing and new drug application, that NDA, controlled standardization, that is a good agriculture practices to be followed. In order to improve quality control of raw materials, good agriculture practice of the raw materials has been greatly increased. That is regulated, was officially used by the government of India, because good agriculture practices is to be used when we export our product to be competitive in the both in the European or in the other countries. When we look into in this point of view, it must be free from pesticides and other residues and the limitations of microbial load. This is comes under when the plant material collected from the wild variety or a cultivated variety. When we look into the cultivated varieties, we have to be much focused on the good agriculture practices to be strictly followed. When we look into the good manufacturing practice, that is good manufacturing practice when it is to be in the pharmaceutical companies, these are it is mandatory to be followed by the GMP guidelines. The GMP regulatory strictly enforced in all the pharmaceuticals, either it may be oral or intraperitoneal or IV, these guidelines to be strictly followed. And the standardization of pharmacological and toxicological studies, not only the GMP, when we look into the laboratory point of view, when you generate the data for the safety point of view, Based on the requirement from regulations for approval of the new drug application, toxicological experiment for the safety evaluation of the drug should be performed in accordance with the GLP standards. When we look into the standardization for clinical use, good clinical practices to be followed. Many hospitals for the drug clinical has been established in accordance with the GCP requirements. In GCP requirements, several the approved clinical investigation of the drugs, that is CID, that has to be followed. That is good clinical practices to be followed. These are the specific standardizations that has to be implemented. Either it may be a good laboratory practice or a good manufacturing practice or a good clinical practices. That good is very much important. When we look into the products in the drug which is available in the market, there are the two pillars which has to be mainly the safety and efficacy is much focused. Because the safety, under the safety guidelines, we'll be talking about the toxicity under these parameters in the toxicology and pharmacology. Because as I have not introduced briefly about the in Lucknow, there are four organizations here. That is of the plant organization. Premier plant organization is National Botanical Research Institute, which we are working exclusively on the plants, not only on the medicinal plants, on the ornamental plants, and chicken kitchen garden and other things. When we look into the efficacy studies, once it is validated, that is Central Drug Research Institute CDRI will be taking, and also the safety point of view, final confirmation we take it from the Indian Toxicological Research Institute ITRC, and then these are the reliable to enter into the clinical studies. Therefore, the preclinical studies, safety and efficacy studies, the pharmacological data taken up to the clinical studies to validate in the human use. As we are much aware in the pharmaceutical point of view, in our routine studies like toxicity and safety, the general toxicity and the reproductive toxicity. Under the general toxicity, we will be talking about the acute studies, subacute studies, and subchronic studies, and the chronic studies. And these are the guidelines to be followed when we talk about the phytopharmaceutical mode of the drug development of the plant origin. We have to talk. We have These are the regulations that the OECD guidelines 420, 14-day study, subacute toxicity studies, 28-day study, subchronic toxicity, 90-day study, reproductive toxicity, that is 1 to 2 years, and each specific guidelines to be followed by the OECD 420, 407, 408, and 443. These are the specific guidelines. Once it is these studies, the data has been generated along with the documented data, we have to submit to the regulatory authority. And these are the specific marker compounds. For example, if you choose a one plant of any plant material, there's the specific how do you do chemical standardization, and the chemical standardization, the four specific chemical markers to be identified, which are the potentially active for the biological use. For example, if you talk about the any single plant, there are the four specific markers to be identified, and it has to be quantified, and qualitative and quantitative analysis to be made, and fingerprinting profile of these markers to make that the product is visible into the market, and identify that product is the commercialized and used, and it has not been substituted or adulterated in the marketing point of view. And these are the routine studies which we usually do in the laboratory. These we know where this is grouping of the animals, route of administrations, whether it is used orally. Most of the acute toxicity studies, the guidelines 420 adopted in the 2001. The duration is 14 days. Age, weight, dosage schedule, urine analysis, histopathology, biochemical analysis, feed and food consumption, uh, feed and water estimation, body weight estimation, and the daily, weekly. These are the data to be generated and is to be provided. Because this we all most of the participants who are involved here are linked here. These are the studies which everyone are doing up to the dose level of 2000 milligram per kg, and these are the data to be recorded. And when you look into the histopathology of the subacute toxicity studies and other data, the vital organs will be taken and taken for the histopathological analysis, and other physiological parameters to be recorded. And when we look into the most of the plants, we grandly take all plants are safe and non-toxic. But there are specific plants which studied to show that it is having teratogenicity effect and the fatal abnormality when injected by the mother in the mother. And these abnormalities, when we look into the thalidomide tragic example of the synthetic drug, these undetected parameters like Seneca, there is a specific constant like pyrrolidine alkaloids like lasocarpine, retrosin. These are the chemical constants that are present in Seneca species, comes under the family of compositae. And these plants is mentioned to be teratogenic. When we are using these plants in the for the academic point of view, in the master's thesis or the PhD thesis, while choosing the plant material, we have to be careful that the while publishing the data, what are the plants that are used for the not only point of the efficacy point of view, the reprints will be used for the other purposes, which not only the efficacy, this has been should be clearly go into the literature review. What are the specific constants that a plant is having as a primary metabolites or a secondary metabolites? These secondary metabolites like pyrazine alkaloids, lysocarpine, retrocerin is well documented. Like at the other plant, when we see Indiophera spatica, that is indospesin is a specific constant. Like Lycotinia species in Solanaceae family, that is pyridine alkaloids. Lobelia species, that is Lobeliaceae family, that is pyridine alkaloids. That is when we look into the Astragalus lentigosus fabaceae family, the chemical constants are unknown, but the plant is reported to be as teratogenic. And Lucenia, Lucophilia, that is mimosin. The mimosin is the specific marker of the biological active constant that causes teratogenicity that obtained from the specific plant, that is Lucania species. And from this, we can understand that all plants are not safe, and the data to be generated and robust data to be created, both at the laboratory level and also to the pilot level studies. And what are the extraction procedures? It is reproducible, and to formulate these extractions or fractionations, and the phytochemical analysis to be made, and the fingerprinting profiles of this plant with the chemical constants to be documented. With this data, this is there not only there are some plants not only causes teratogenicity, there are some other plants which causes liver damage. That is nothing but hepatotoxicity related to the herbal remedies. These are the herbs of the tags are like Crotinaria symphoticum. That is a there is a pyrrolizidine alkaloids which causes liver damage. That is causes hepatotoxicity. That is mentha puligium monoterpene. It causes hepatotoxicity. Cassia angustifolia, anthrin chemical constant that is responsible for causing hepatotoxicity obtained from the Cassia angustifolia. That is when we look into the other species, that is a chemical constant, that is saffrole, that is obtained from a saffrous albidum, that is also causes the hepatotoxicity. That the one of the piper species, that is piper methyseticum, it causes hepatotoxicity, that is liver damage, and increases the level of the other biochemical markers. This has been identified in the liver biochemical markers like SGPT, SGOT, albumin, and other parameters. And how do you project these? How do you extract and how do you fractionate these constants? The bioactive activity fractions to be identified. When we go into further extraction, use it for the different solvent extractions, and you get the different fractions. And these fractions should be subjected to for the biological activity. And if you identified that the F2 fraction is activity as shown biological activity, and F1 and F3, F4 is to be rejected, there is no activity. F2 has to be subjected for further fractionation for the structural and spectral analysis through the column chromatography, HPLC, HPTLC, LCMS, and NMR. And other data in today's scenario, we can get the spectra completely on the profile of the fraction of this bioactive fraction, which has been and the structure to be identified. And this is one of the example which we have taken in our laboratory studies, that the different ulcer models which we have designed, like surgical method, that is 4 hours pylorus ligation method, and restrained stress induced ulcer, that is cold restrained stress ulcer, and alcohol causes ulceration in the human and also in the animal model, that is ethanol 1 ml at 200 grams that causes ulcer, and the drug induced ulcer, that is aspirin 200 milligram per kg orally, when you give to the in empty stomach, it causes gastric ulceration. There are specific parameters which is to be identified both at the gastric mucosal and serosal end of the stomach. There is the defensive parameters like mucin content, whereas offensive parameters like hydrochloric acid, like pepsin, and this increases the ulceration in the stomach. There is the blanket of the stomach at the glandular portion of the stomach, at the defensive, which the mucin content and the mucin content thickness of the stomach in the inner as a blanket to be quantified. And how before entering into the animal model studies, we have to understand what are the specific component of marketed products which we take as a benchmark or a positive control. When we talk about the product that we want to compare with the proton pump blockers like omeprazole, that is H plus K plus ATPase, then we have to take for the specific positive control as omeprazole. If you want to compare with the H2 blockers like ranitidine or cimetidine, we'll be taking this positive control of ranitidine or cimetidine, and we will compare our the plant extract or a fraction with this specific synthetic molecules, whether the plant extraction or the fractionated compound, or identified the specific marker compound from the specific plant of the biological activity, and and the structural analysis, and these are the backup of the chemistry. We give the clear indication, sorry, and these are the, and based on the modern synthetic drugs which are which we study in the pharmacology point of view, those products which are available as H2 receptor blockers or a proton pump blockers as taken as a reference compound, and compare with that data along with the treated groups, along with the group which has not been untreated. When we look into the like aspirin, and when we design the models for in acute ulceration and chronic ulceration, there is a COX inhibitors, COX1 and COX2 in the healing of the gastric ulcer play a significant role here. That is intact the gastric mucosa. These are the COX1 and COX2. After it is synthesized from the arachidonic acid, that is an enzyme induction, that is constitute the enzyme activity in the physiological prostaglandin. Prostaglandins are cytoprotective, and these how these prostaglandins protect the ulceration in the stomach, and what are the pathological prostaglandins are the like how it causes inflammation, how it causes pain, acceleration of ulcer healing, that is causing to the enzyme induction through the COX2, and these comes under the category of the NSAID, non-steroidal anti-inflammatory drugs. Growth factors and the gut peptides and exogenous prostaglandins, glucocorticoids. These under the class of the drugs which under the classification of the exogenous prostaglandins and glucocorticoids also plays a significant role in the on COX2. On this healing, how these COX1 and COX2 inhibitors, how these are being used in the healing of the ulcers by inhibiting the COX1 and COX2. And when we look into the prostaglandin like misoprostol, when we look into the H2 receptor blocker like cimetidine and ranitidine, when we look into the other things like paratels, like proton pump blockers like omeprazole and proclomide, the gastrin receptor blockers, these are there. In the initial stage, we used antacids like a gastric acidity. In the secondary phase, we used the H2 receptors. In the third phase, we used the H plus K plus proton pump blockers, that is omeprazole. Based on this, we can when not only the data generated in the preclinical studies, when we subject keeping in mind in the clinical applications of this fraction, when it is commercially available, in which stage, and whether it is used in the acute stages or the chronic stages, the product to be well aware, the product and the patient should be well aware in which stage when should be used, that has been prescribed by the physician. These are the when we look into when we look into the concept of the prodrugs itself are not active. When it is metabolized, the metabolic products are active. Like one of the example is the omeprazole. Omeprazole itself is not active. When it enters into the bloodstream, into the parietal cells, in converted into the specific inhibitors which act and acts through the H plus K plus ATPase. These metabolites are specifically acting and inhibiting the H plus K plus ATPase. Then the concept of the prodrugs has to be kept in mind. What is the prodrugs itself is not active. The metabolites are active. Therefore, what are the metabolites and the omeprazole, where it is metabolized, what are the liver function tests, and make it as an active compound, what are the steps involved, and how the sulfonide intermediates and the enzyme inhibitor complex has been formed, and how these inhibitors inhibited the parietal cells. And we're not only looking into the gastric ulcers of acute and chronic, we also looked into the concept of the model of the gastroesophageal reflux disease we have designed in our laboratory. Because not only looking, this is most of the thing they are confused with the heartburn, heart attack. It is more reflecting towards to the heart. The gastroesophageal reflux disease is a heartburn, but doesn't correlate to the heart. It is a reflex esophagitis, that is the ligation at the forestomach and the glandular portion, followed by the pyrosis-induced esophagitis. And initially, after the 1 hour of the pyrosis ligation, forestomach and the corpus ligation, the pyrosis ligation, and 6 hours, and go for the treatment and observation. And this paper has been published in the European Journal of Pharmacology. In the continuation of studies, these plant extracts and the fractionations, and these extracts has been subjected for the extracted, and surgery has been made, and animals has been observed for the ulceration in the esophagitis, and what is the gastric volume, what is the index of that, and based on the dose response studies, and what is the gastric volume, and this data has been generated, and based on the acid output, and also the pepsin output. These are the acid and the pepsin are the offensive factors. And where they not only this, there are the free radicals like lipid peroxidation. Lipid peroxidation inhibits the the effect of lipid peroxidation during the course of the study of the surgery. And what is the effect of these plant extracts or the herbal products on the inhibition of the lipid peroxidation at the esophageal end of the stomach? This has been studied and showed a very significant effect at the 300 milligram per kg of Euphorbia aerata. And when we look into the enzymatic antioxidants and non-enzymatic antioxidants, the enzymatic antioxidant like catalase, superoxide dismutase, reduced glutathione, and reflex esophagitis. When we look into the compare the data with the esophagitis with the control group, there is a specific enzymatic antioxidants have been reduced. When we treated the extracts with the different plants and compare with that of the omeprazole, that OME, that is H plus K plus ATPase, there is a significant promising activity having similar to that of the synthetic compound like Euphorbia aerata. In this composition, we went in this line. We have studied several plant extracts, and we filed a patent, and we published it for the data. After the publication, we went for the in depth of the studies, and we went for the publications. And a few students has been registered in this line, and more than 5 students has taken PhD in this line of experimental data. And the data which we have generated, not only on the esophagitis, we used it for the gastric ischemia perfusion. The ciliary artery was included for 30 minutes, followed by perfusion, which it has been published in Journal of Pharmacy and Pharmacology. And the reflex esophagitis, how we used the new model which we designed for the gastroesophageal reflux disease. And the several patents in the not only in the Indian scenario, we taken the patent from the United States, which the patent has been granted, and the patent has been granted, and the product has been developed, and we made publications in the European Journal of Pharmacology and also the Journal of Pharmacy and Pharmacology. What else the data we have generated during the course of this time in the development of the product. And there are specific quality evaluation. How do you, when we look into the pharmacognosy point of view in the pharmacy line, that the HPLC as a tool for the quality evaluation and standardization of the plants and herbal formulations. As we know, Trifla is a combination of 3 plants. That is HPLC PDA protocol has been developed, and the qualitative and quantitative estimation has been made, and the specific markers has been developed, and the data has been published in the phytochemical analysis, that the gallic acid content of the individual and also the final products has been made by the HPLC. And not only on the gastric ulceration, we also worked out on the gastrointestinal disorders, and which in the different animal models we have worked out. And the eagle marmalade, that is the bale, which has been mostly as a fruit we have taken, and this has been published in this line. The publication has been made, and followed by initially we file a patent and the product has been developed. And this is one of the product based on the studies and the strength keeping in mind of the regulations. There are several few products I'll be showing here. This is the anti-ulcer product that neutralizes the excess acid in the stomach, used in acute and chronic gastric ulceration, used in healing and curing of ulcers and in internal bleeding. And this is the herbal products in the nutraceutical concept, fortified with herbs, reported in Ayurveda to have a health product, protective and provide better nutrition. And the drink is provided for it's said to be as a hepatoprotective. And there is the herbal fermented health drink. It is attributed to the hepatoprotective and antioxidant properties. It provides instant energy and combat fatigue. These are the in other sector which will be working on the cosmeceuticals point of view. That for example, if the colors, the different flowers are having the anthocyanin pigments, which we, and other pigments which we seen on the flowers. Those pigments of the colors has been isolated, and the colors has been taken and studied for the different allergic reactions. And those colors has been taken for the antimicrobial, antioxidant, and anti-inflammatory, and has been those colors has been taken for the development of the product. And one of the product is the this is the lipstick which has been developed, and the technology has been transferred to the Ayur herbals. A novel herbal formulation for the treatments of cuts, burns, and wounds. These are the because this herbal formulation has an antimicrobial and antifungal properties and relieve the pain and inflammation, and used for the regeneration of the dead cells. It has been covered under the IPR and has been protected, and and it's commercially presently the technology has been transferred. And the biosynthesis of silver nanoparticles from trichoderma, that is a wool healing activity which one of the which of one of the antimicrobial department has developed and studied for the parameters like other wound healing effect of this product, like which contains the trichoderma isolates, and it shown a significant activity, and when they compared with the betadine and soframycin. This is one of the major product which has given more than 7 crores of royalty to the institute. The joint product developed by the NBRI and also Central Institute of Medicine and Aromatic Plants. This because I am in a National Botanical Research Institute, and one of the other institute that is there within our umbrella of the CSR is Central Institute of Medicine and Aromatic Plants. This is diabetes mellitus. It is a complex metabolic disorder resulting from either insulin insufficiency or insulin dysfunction. Based on the traditional knowledge, scientifically validated herbal product has been developed for the management of diabetes. These are the silent features. How do we process it, and what are the requirements, and how do we follow these regulations to bring into this experience and the studies and the protocols and the regulations both on the in the scenario of the Ayush mode. The product has been developed and the product has been launched, and it was commercialized to the Amil Pharmaceuticals, and the product is visible on the bench in the pharmacy departments in the market. And these trade name they given as a BGR34 because it contains 34 chemical constituents biomarkers in this plant. This is the other product like herbal cough formulation dosage form. These how these regulations has made us successful to come into the market to access the industry. And there's one of the product like herbal cough formulation dosage forms. It is a syrup. The technology has been transferred to the Torah herbals. It is a combination of 5 indigenous medicinal plants. It has been protected and covered under IPR. And my other product is nutraceutical jam, a herbal jam rich in nutrition. The product is help for the mixture of minerals and fruit pulp. As I said in the regulations, it is already not only the plant materials or the extracts or the fractionations that is mixed with the minerals and the fruit pulp. The product is purely natural and herbal in nature. The technology is important specifically for the kids of health, and the technology ready for commercialization. And presently, the technology has been transferred. This herbal black dye used in the beautifying the hair and its applications, because the dye that is extracted from the plant materials and it is fortified, and these studies it needs more advanced studies for the stability point of view, which we are looking in depth presently. And these most of the leads that are obtained from the indigenous community like tribes like Kani tribes, which most of the share of the royalty which we get, and also share to the who disclose the leads and may make us to be successful. This is one of the model, that is Pushpangam model for benefit sharing, and it is executed in 1995. That is Kani tribes and the institute at the 50% royalty. That one of the product is drug Jivani was released by the technology transfer to Ayurveda Protocol, and one of the product also is in Coimbatore. Thank you very much for giving an opportunity. If any questions, I'll be happy to answer resolve the issues which you are facing during the course of the R&D which you are working in your laboratory. This is one of the presentation I have made. I have to make the other presentation. The other presentation we have made on the antiviral studies, which the other regulations I'll be talking about, the handling the coronavirus, SARS or a dengue virus, how to handle, and what are the studies we have made. That I'll be sharing in the next presentation, madam. If any questions, I'll be happy. Thank you, madam, for giving an opportunity and be with you here.
Sir, for your wide elaborate talk on phytopharmaceutical drug, you have used the term particularly.
Yes.
So you were talking about pharmacological evaluation, safety studies of herbal drugs.
Yes.
So your concern is that FDA, new drug applications, IND submissions, these require safety and quality aspects of herbal drugs.
Right.
And when we look at your talk, sir, you are wonderful and excellent. You have published around 150 original articles. I think you are always 24 hours involved in research, I think. So many products, all categories of products, jams, everything. You have shown colors, hair dyes, therapeutics, whatnot. You have shown so many products of your own, and you have got patents also, 102. So excellent portfolio, sir. A lot of things we have to learn from you. Should make you as a role model and forward in our careers. So you have told about FSSAI guidelines, sir, and you said that 400 botanical plants are listed along with their permitted use, and you have talked about 187 ingredients. Means their safety protocols, everything is ready. We can use them. So you have used pure. You said there are pure natural products, extracts with marker content, everything is standardized. So,
Yes.
and you have also speak about teratogen plants, hepatotoxic plants, and you explained about GERD, animal screening model, and HPLC as a tool for quality evaluation and standardization of herbal formulations, plants, and whatnot, sir. Everything you have covered uh, along with standardization, herbal safety issues. So thank you very much, sir. Thank you. And I think you can proceed with your next talk.
Madam, we can have a break of 5 minutes.
Yes,
Yeah,
sir.
because I have to
Okay,
look.
sir. Yes, sir. Yes.
Yes. Thank you.
I request all the delegates to have a tea break of 5 to 10 minutes and we rejoin the session.
Yes.